afternoon, everyone, and thanks to the organisers for this opportunity to present Opthea's program developing OPT302 for the treatment of wet age-related macular degeneration. So Opthea Limited is a public company. We're listed on the Australian Stock Exchange under the ticker symbol OPT, and we are developing a lead compound called OPT302, which targets two alternative members of the vascular endothelial growth factor family, VEGF-C and VEGF-D. So VEGF-C and D are important mediators of neovascular functions. They induce angiogenesis as well as vascular permeability, but can also mediate non-neovascular functions as well, such as inflammation, all of which are very important for very much hallmarks of wet AMD as well as other ocular diseases. We're developing OPT302 as a combination therapy with the existing anti-VEGF-A agents in order to mediate a more complete blockade of this very important VEGF receptor family, but we're also potentially complementary to other novel therapies that are currently in development as well. We have an ongoing phase 1/2A clinical study that's being conducted under an IND in the United States. We are currently recruiting into the phase 2A dose expansion cohort and have recently reported top-line data from our phase 1 clinical trial. So the development strategy for OPT302 is to address the suboptimal response that's associated with long-term single-agent therapy with VEGF-A inhibitors, including the suboptimal improvements in visual acuity and the persistent retinal fluid, which is observed after long-term treatment with a VEGF-A inhibitor. Resistance to VEGF-A inhibitors may indeed be related to other family members. The family goes beyond just VEGF-A, and in fact VEGF-C and D share overlapping signalling pathways with VEGF-A, as well as signal through an independent pathway through a separate receptor. OPT302 targets VEGF-C and VEGF-D through a trap mechanism. It potently blocks both, blocking to the receptors, and can also block the consequential upregulation of VEGF-C and D, which can occur when VEGF-A is inhibited. Used in combination with a VEGF-A inhibitor, we can more completely block the VEGF pathway, prevent these elevations in these alternative family members, and target an incomplete response to VEGF-A inhibition. This concept of VEGF-C upregulation when VEGF-A is inhibited is demonstrated here, with the aqueous levels of VEGF-C significantly elevated following 1 and 2 months of therapy with Avastin following intravitreal injection in wet AMD patients. The design of our phase 1/2A clinical trial is shown in this slide. You can see here that we're conducting the trial in essentially 2 parts. The first is a phase 1 dose escalation study of 20 patients in which OPT302 is combined at 3 sequential dose levels in combination with Lucentis. And in that phase 1 trial, we've also enrolled 5 patients to receive the monotherapy of the drug as well. The phase 2A clinical trial is currently still ongoing. We're recruiting patients. That part of the study will recruit an additional 30 patients randomised to either monotherapy or the high-dose combination cohort. The phase 1 clinical study recruited 20 patients, the majority of which presented with occult lesions. 17 of 20 presented with occult lesions, and we recruited 1 patient with a predominantly classic lesion and 2 patients with minimally classic lesions. Each patient in the study received 3 intravitreal injections of OPT302, either alone or in combination with Lucentis over a 4-week period, with a week 12 follow-up 1 month after the 3rd dose. The majority of our patients that were recruited in this clinical study were difficult to treat patients. They had received prior anti-VEGF-A therapy and had shown a sub-response to that therapy, and in addition we recruited an additional 30% of patients that were treatment naive. We successfully met the primary safety objective of this clinical study. As you can see here, we saw no drug-related serious adverse events and no evidence of drug-related immunogenicity. Overall, 16 out of 19 evaluable patients maintained or gained vision from baseline to week 12. No patient lost more than 3 letters in the study. Looking at the combination therapy across all patients enrolled that were treated with the combination, including both naive and prior treated patients, we observed a mean visual acuity gain from baseline at week 12 of 8 letters overall, and a mean decrease in central subfield thickness from baseline to week 12 of 91 microns. Looking only at the naive patients that we recruited and treated with the combination therapy, we recruited 4 patients in this phase 1 clinical study and observed a mean gain in visual acuity from baseline in those patients of 16.5 letters. together with a reduction in the central subfield thickness of 214 microns, which was a 42.7% reduction in the retinal thickness of these patients that were treatment naive and treated with the combination. In our prior treated patient population, we had 10 patients treated with the combination. The mean change in visual acuity at week 12 from baseline was 4 letters in this very difficult to treat patient population. And we saw a further reduction of 11% in the central retinal thickness of this prior treated patient population, many of whom had received extensive prior therapy, some up to 55 prior injections. In our monotherapy cohort, 3 of the 5 patients did not require rescue therapy with Lucentis. Of the 2 patients that were rescued, they were rescued relatively early. At week 12, both had indeed continued to lose vision compared to baseline. So they indeed were very difficult to treat patients, and Lucentis therapy did not achieve an improvement in visual acuity in those patients. At week 12, in those patients that did not require rescue therapy, the mean visual acuity gain in those monotherapy patients was 3 letters from baseline. 1 patient experienced a 6-letter gain and a mean increase in retinal thickness of 18 microns, which we believe is clinically stable. So just to finalise and summarise our program, we have a program which is a trap molecule targeting 2 alternative members of the VEGF pathway that has the potential to address a large unmet medical need for wet AMD, given that the current treatments only target VEGF-A. We've met our primary safety objective of the phase 1 study, and we've provided evidence of clinical activity from this 20-patient clinical study. Those patients that were treated with the combination showed mean visual acuity gains and retinal thickness improvements that were equal to or superior to historic 12-week data with Lucentis alone, despite having a very heavily pre-treated patient population and patients with a high proportion of occult lesions. In naive patients, our data suggests that there is a meaningful additional VA gain and a reduction in CST over and above what we would expect with Lucentis alone, and we saw a stabilisation in the monotherapy group. The totality of our data warrants advancing OPT-302 combination therapy into a phase 2b randomised controlled trial, and the planning for that study is underway now, and we expect to initiate that in 2017. Right now, we're actively accruing into our dose expansion cohorts of the phase 2a clinical study. Thank you very much.